Welcome to another lecture by MedicoMedics, Learning Made Easy, Genetics Chapter 7, Population Genetics. So in this lecture, we are discussing population genetics, talking about genetic variation, the Hardy-Weinberg Principle, factors affecting genetic variation, continuing with gene flow, natural selection, application, and end with a summary. Now, population genetics is the study of how genetic variation is distributed and changes over time within a population. The goal is to understand genetic diversity and to analyze evolutionary forces shaping populations. Why is it important? Because it helps in disease studies, in conservation biology, and understanding human evolution. So population genetics looks at how genes are spread across groups of people, animals, or plants, and how they change over time. So what are some sources of variation? Well, it can be through mutation, where new alleles are introduced. Recombination, which shuffles genes during meiosis. And gene flow, the movement of genes between populations. Then we have the concept of genotype versus allele frequency. So genotype frequency is the proportion of individuals with a specific genetic makeup. Allele frequency means proportion of a specific allele in the population. So genetic variation comes from mutations, gene mixing, and movement of individuals, creating diversity in population. An example of this could be in a population. If 70% of people have allele A, capital A, whatever that is, and 30% have allele A, the allele frequencies are 70% and 30%. So now let's see some practical calculations. So we have the Hardy-Weinberg Principle. And it is a mathematical model used in population genetics to predict that allele and genotype frequencies in a population will remain constant from generation to generation, provided that no evolutionary forces such as natural selection, mutations, migration, genetic drift, etc., act on it. So it kind of represents a genetic equilibrium. Now, for a population to be in this Hardy-Weinberg equilibrium, some conditions must be met. So we make these assumptions. First, large population size. So this minimizes genetic drift. Then we have random mating. So no preferences for specific traits. That's another assumption. Then we have no mutations, so no new alleles introduced. No gene flow, meaning no migration in or out. And no natural selection, so no allele has a reproductive advantage or a survival advantage. So these are the basic assumptions. Then we have a way to use this to identify how many percent of individuals are a certain are heterozygotes. So we have the equation here, right? P squared plus 2pq plus q squared is equal to 1. So p squared here is the frequency of homozygous dominant or homozygous dominant, so AA. 2pq is frequency of heterozygotes, AA, and q2 again, but for recessive, so AA. And here is an example of a calculation. So if P, the dominant allele, is 0.6, that squared will give us 36%. And if Q, the recessive allele, is 0.4, that will give us 0.16%. So 2PQ would give us 2 times 0.6 and 0.4. That will give us 0.48, meaning 48% of individuals are heterozygous. And this principle helps scientists study whether a population is evolving by comparing real-world genetic data to expected frequencies under equilibrium. Now, what are some factors affecting genetic variation? Well, we have mutation, where there is an introduction of new alleles. We mentioned before gene flow, so the movement of alleles between populations. Genetic drift, so random changes in allele frequencies, especially in small populations. And of course, natural selection, so survival and reproduction of individuals with advantage traits. So for example, if you look at genetic drift, so in a small village, a rare allele might disappear by chance if no one with it has children. So genetic drift are random changes in allele frequencies in small populations. The effect would be reducing genetic diversity and increase chances of losing rare alleles. The types are bottleneck effect, so a population is drastically reduced, leaving limited genetic diversity. Then we have founder effect, so a small group establishes a new population with limited variation. Example, bottleneck, so a natural disaster kills most of a population, leaving only a few survivors with a narrow gene pool. Gene flow is the movement of alleles between populations through migration. The effects: increasing genetic variation in a population, but reduces differences between populations. So for example, a group of individuals migrate to a new region and mixes their genes with the local population. Natural selection. The definition is differential survival and reproduction of individuals due to advantageous traits. Types could be stabilizing selection, so favors the average trait, like human birth weight. Directional selection favors one extreme trait, like faster cheetahs. Disruptive selection favors both extremes, not the average. So for example, beak sizes in finches. So example here is antibiotic resistance in bacteria is an example of directional selection. So natural selection essentially is like nature choosing the best tools for survival. And so these tools, these traits, become more common. Some applications of population genetics. So in medicine, for example, understanding genetic predispositions to diseases. It helps identifying populations at risk for inherited disorders. In conservation biology, it helps preserving genetic diversity in endangered species. In human evolution, helps us trace migration patterns and ancestry. A medical example could be sickle cell allele persists in population where malaria is common because it provides resistance to the disease. In summary then, genetic variation is influenced by mutation, gene flow, genetic drift, and natural selection. The Hardy-Weinberg Principle provides a baseline for studying populations, and applications range from medicine to conservation and understanding human evolution. So, all in all, population genetics looks at how genes change groups over time, helping us understand diseases, evolution, and even how to save endangered species. And that's the end of Chapter 7. Continue now to Chapter 8.